So let's talk about fun stuff, about HIV. Just going to leave this here. So HIV, or human immunodeficiency virus, is the single-strand RNA virus. It's enveloped. And I'm going to draw an infection cycle, which is fascinating. So, actually from that same unit, from this unit that we're talking about, what do viruses need to attach to the surface of the cell? Anyone remembers? What do they need?
Receptor?
Yes, thank you very much. They need a receptor. So this is our virus, okay, schematically, and it needs receptors. As a matter of fact, HIV needs two. So it needs one, and it needs another. Again, this is schematic, right? So one of them is always CD4 molecule, and yes, it is CD4 from T cells, right? So this virus infects CD4 T cells. It can infect macrophages, but for our intents and purposes, we're going to focus on CD4 T cells. Deal? The second so-called core receptor, CCR5, it's a cytokine receptor, more famous. Less famous is CXCR4. Now, I want to be very clear. I don't care much about CXCR4, but for CCR5, it can carry a deletion. Now, this is the great opportunity to use a real-life analogy. Essentially, receptors are the locks or keys, call it whatever you want, for the virus to enter the cell. Does that make sense? And obviously, in order to open the doors, keys must be perfect. Does that make sense? What if one of the keys, CCR5, is missing a substantial chunk of it? Do you think door can be opened? Exactly. There's a very small percentage, about different genomic studies, they suggest differently, but about 0.1% of people of Northern European descent carry Delta 32 mutation in CCR5. Don't memorize all those stupid numbers, please, names. It's going to go away unless you work with it, okay? The point is, there is a fraction of people who are genetically resistant to HIV infection. Okay? Again, we're talking about clade 1, which is clade 1 of HIV, which is most common in the United States. They can be infected with different types of virus, but it's super rare. So generally speaking, we have a proportion of people that are very, very unlikely to develop HIV. Does that make sense? So let's say virus gets into the circulation, infects CD4 T cells. So what happens next? Well, virus gets into the cell in the process of, so this whole process often is referred to as attachment and fusion, and then it releases its single-stranded RNA. Now, do you remember the process of gene expression, like the steps, the cookbook thing? Remember? So single-stranded RNA. What serves as the template for single-stranded RNA synthesis in normal cellular gene expression? Anyone remembers? You want to say it, say it. If it's wrong, it's fine. Oh, come on. So what are the steps? Okay, line me the steps of gene expression. What do we start with? Transcription is the process of using what for making what? In transcription, what happens in transcription? DNA? DNA is used to synthesize mRNA. Does that make sense? You wanted to say this, you just hesitated. So, single-stranded RNA is the product of DNA transcription. Now imagine you take this step and run it backwards. So you take single-stranded RNA and use it as a template to produce double-stranded DNA. Does that make sense, that statement? So this process here is called reverse transcription. And not surprisingly, the protein that facilitates it is called reverse transcriptase. Often the acronym is used for it, RT. So now what you're going to do with double-stranded DNA, the next step is also pretty cool. So this double-stranded DNA is being sent into the nucleus where it's incorporated into cellular DNA. Does that make sense? In the form of provirus. This process of integration is facilitated by the enzyme surprisingly called integrase. Okay? Does that make sense? Now, what is important, what is crucial to understand here is once provirus is integrated into the genome, it's there forever. That's it. You can't get rid of it. Make sense? I mean, unless hypothetically you get rid of all CD4 T cells in a human body. But it's virtually impossible. Okay, we're good? Now what happens next? Well, now we get a normal gene expression. So this fragment, this provirus, serves as the template in transcription to make viral mRNA, which is then used in the translation to make viral proteins. So as you can see, all pink color refers to the virus. Does that make sense? And you have multiple. Now, here's something. This mRNA that I'm referring to and this single-stranded RNA are essentially the same. Be clear? I mean, they're not like literally, there's some slight differences, but you get the gist. So at the end of the day, proteins and RNA get assembled into new virus particles. This is some big-ass virus particle. And the virus particle gets out of the cell. Now I want to make sure that we'll hear on the same page. This step, when it gets out of the cell, it requires some extra processing. It's called proteolytic processing. Okay? Which is facilitated by the enzyme protease. So let me kind of make it in one smooth way. Virus binds to the receptors, makes it in the cell. Single-stranded RNA is converted to double-stranded DNA by reverse transcriptase. Double-stranded DNA is integrated into human genome of CD4 cell by integrase. And then this provirus serves as the template for both expression of viral genes to make viral proteins and to the synthesis of viral genomes, the RNA genomes. Everything gets packaged neatly in the particles, and when particles get out of the cell, that process requires proteolytic processing. Okay? And then this mature particle is ready to go and infect another cell. Okay, so what do the symptoms look like? Upon initial infection, it's usually some fever, maybe a bit of night sweats. And of course, your CD4 T cell counts start to drop. Does that make sense? So usually the first period of virus infection, the HIV infection in humans, is referred to as latency. Although, strictly speaking, check this out, it's not latency in the classical terms. You can detect the virus in the blood. Basically, it's the delicate balance between the immune system and the virus. But here's the problem. Virus, by replicating in the CD4 T cells, kills them. So levels of CD4 T cells, they constantly decline. Are you following me? And at some point, and this point, this slow kind of what's called latent phase may last for years. If you look at the poster child of HIV AIDS, Freddie Mercury, the singer for Queen, he apparently got infected sometime in 1986, and his symptoms finally showed up like 2.5 years later. Okay, so it takes a while, but eventually the number of CD4 T cells drop precipitously, so that component of the immune system is gone. And you remember that if you don't have CD4s, your B cells suck, your macrophages suck, your CD8 T cells suck. So people start contracting secondary infections, which can vary from CMV-induced hepatitis to protozoan infections. It's known, for instance, that some protozoan infections that may not even affect a healthy person establish persistent colonization and result in chronic diarrhea, for instance. Fungal pneumonias, Kaposi sarcoma, herpes virus, skin cancer, meningitis, fungal meningitis, amoebic keratitis, all kinds of terrible diseases. Does that make sense? People basically die from the secondary infection due to the immunosuppression. Okay? So, a few questions that we should ask. Is there a vaccine? No. And it's very sort of, this virus kind of defies the concept of vaccination. Because for the vaccination, what do you need? First of all, you need a more or less stable pathogen. HIV is nothing but genetically stable, mutates really quickly, changes antigenicity. So creating like a broad range vaccine is so far not helpful. Second problem. Let's talk about, use different example. Let's talk about COVID for a second. So I've been vaccinated, you all been vaccinated, right? So let's say I'm vaccinated, I get infected. Let's say my immune response fails to, say I didn't respond well to the vaccine, so I have very subpar immune response, I get sick. Say I'm not dying from this, I just get sick. I spend a day, 2, 3 at home, feeling lousy. I get better, I clear the virus. Does that make sense? I can go back to normal life. I don't have COVID anymore. Here's the thing. If the vaccine fails to prevent initial infection, even in 1 out of 1,000 vaccinees, this person will harbor HIV forever. Does that make sense? Like, just one miss and the person is infected for life. It's not like they're going to get better. You see what I'm saying? This is the infection for life. So that's kind of a trouble. So vaccine efforts actually focus on developing something that would help to control the virus in people who are already infected. Does it make sense? And one of the reasons why vaccine research in HIV is kind of sluggish, we have phenomenal treatments. Okay, really phenomenal. There are drugs that target attachment and fusion. There are drugs that target reverse transcription. There are drugs that target integration. And there are drugs that target proteolytic processing. Probably have like 3 dozen drugs against HIV. And these drugs are used in a combination, of course, to avoid the development of resistance. And they're so good that WHO classifies HIV as the chronic disease. You know what else is chronic disease? Diabetes and hypertension. So basically WHO says that with proper management, HIV does not impact the life expectancy. Make sense? Of course, it's all about money. So therapies are pretty expensive. Okay. Any questions so far? And look at Magic Johnson. I'm gonna die and this dude's still gonna comment about basketball on ESPN. Now, transmission routes. Depends on the country. To a large extent in the US, it's mostly intravenous drug use. So the best way to prevent HIV among intravenous drug users is to give them syringes. It's been shown multiple times. Just, that's it. Blood donations, blood transfusions. In the US, practically gone. All blood is gone. All blood is tested. Sexual transmission. So sexual transmission can occur as well as IV drug use transmission can occur if the virus is present in the blood. So this kind of represents another very, very sensitive point. People who religiously take what's called, I'm going to put this acronym so you know for your future clinical experiences, highly active antiretroviral therapy, or HAART. So people who are in the HAART, most of them don't have detectable levels of virus in the blood. They're still infected. Does that make sense? You just can't detect the virus in the blood. And those people can't transmit. So in case of unprotected sex, they can't transmit. Okay? Cannot. Yes, cannot. And that's actually a big problem because those people want to not notify the sexual partners that they are HIV positive, because now you must do that. Okay? But it's, I'm not touching it with 6-foot pole. Okay? It's very, very contentious issue. Anyway, sexual transmission. Anal sex is more risky than vaginal sex, obviously, and I'm talking about unprotected. Okay? Yes, condoms are 99% protective, 1% accounts for unfortunate accidents. So basically, the virus can be transmitted from penile mucosa to rectal mucosa and vice versa. Okay? Also, traumatic nature results sometimes in a blood exposure, so that contributes to higher risk of transmission during the anal sex. Vaginal sex, penile mucosa, vaginal mucosa, between them. Either way. You see what I'm saying? It can be an infected male, uninfected female. It can be infected female, uninfected male. Doesn't matter. Oral sex. Transmission between the mucosa is not possible. So virus cannot cross oral mucosal layer. Does that make sense? And virus is not in the saliva and not in the mucus in the mouth. So all the cases of transmission of HIV during oral sex are due to the wounds in the mouth, bleeding gums, poor hygiene. That makes sense? My favorite, of course, is kissing. There were cases of HIV transmission during kissing. In the report that I read, the kissing was called violent. So basically people were just biting each other. So again, it's blood exposure. Okay? Vertical transmission. Vertical transmission is quite interesting. It's a very common route for HIV transmission in the developing countries. In the United States, I didn't check the latest numbers, but at some point it was in low double digits per country. So like 26 cases. Because HAART is extremely effective and safe. Pregnant women can receive highly active antiretroviral therapy, which prevents the transmission to the fetus. So it is absolutely 100% possible to treat pregnant women who are HIV positive, and they will give birth to an HIV negative child. That makes sense? Now, when just a little note, this pandemic, the one that we kind of at the very, very long tail of now, it's going to be over at some point. HIV pandemic has been around since like 1990s, and it's going to be around for many, many, many more years. Okay, just put things in perspective. Questions? Okay.